About quantifying integrating molecules for a more precise medicine. And as Flavia mentioned, I am a biologist, so I did not have any background in computational science or mathematics or physics. So I had to learn in my PhD because the bioinformatics in the lab did not have time to help me. So because I'm Japanese, I say, I'm gonna learn this, and then I enjoy and became my passion. So I'm going to show you some projects in my lab that is not deep immunology and biology, because I know that there are people here that are not on the biological field. So I try to please everyone, but of course some will enjoy more than others. Just a little context, I became biologist because of the Human Genome Project. Probably you guys were not even born in 1990. In 1996, I was in high school, and then I heard about the Human Genome Project, and then I decided to do biology. And when I went to the University of São Paulo for biology, like I did an internship, and I started my work on molecular biology. So I did this, guys. I was literally reading DNA bases by hand, you know, like saying like, okay, A, C, G. And imagine a human genome with 3.2 billion base pairs. How could you do this 40 times for each one of these 3.2 billion base pairs? So that we definitely need some better technologies to really sequence the human genome. And I saw all these things happening because in 2002, I was in my PhD when we were analyzing microarrays. At the time was the big things, like 3,000 things at the same time. We're measuring RNA, like a lot of... My PhD, for example, was analyzing 6 million reads. At that time, nobody had this, heard so many sequences. And now, a single lab can do this in a few days, right? So I witnessed when the Human Genome Project that cost $2.7 billion and 14 years to finish one genome, now you can do it in a few days and for less than $1,000. And that's because there was a lot of revolutions in the robotics, technology, computers, internet, lasers. And then molecular biology that used to deal with thousands of features, then at that time became the high throughput biology that deals with millions of features. Now you're measuring the activity of 20,000 genes across several samples, humans samples or tissues. And now we are in the phase of systems biology, which deals with several billion features. And now you understand why it's so important for you guys to learn quantitative biology and why I am interested in see who is here, because I may see you in the future, and I hope so. So I did this in my social media just to show that when I was in my PhD, we were representing high throughput sequencing, high throughput image data like this, like heat maps and gene networks, and now it's very, very fancy and beautiful figures. If you look at immunology, which is my field of expertise, every single low throughput technique, the conventional technique that people do in the lab, now there is an equivalent, which is a high throughput technique. So you can measure all the proteins using mass spectrometry. You can do editing of the genome in few months. You can analyze the activity of all the genes, the immune repertoire, and also instead of measuring the 14 markers, you can measure hundreds or dozens of markers using CyTOF. And that project, that one genome project, now became the 1,000 genome project, and then the 100,000 genome project, and now the 1 million genome project. In China, even have a bigger project than this. There are several other nice projects, such as ENCODE, that after you read the DNA, you have to try to decode what the sequence means. And also the Human Cell Atlas, which is going to measure the activity of 20,000 genes in all the trillion cells of your body. So they wanted to create an atlas of expression in every single cell of your of your body. And I'm happy to be part of the Human Cell Atlas Brazil, which is doing single cell things here. In the network medicine is how we apply everything we learned in the lab to human health. And why network medicine is important? Because when we are infected by a pathogen or when we receive the same vaccine, we may have completely different outcomes. Some people will be fine, some people will be sick or even die from the disease, right? The same disease can cause completely different outcomes. And the reason for this is that we have a unique immune system. We have different genetic backgrounds. One is male, one is female, one is old, one is young, one has a malnutrition, other one has stress. All this impacts your outcome. And of course, if you're going to use high throughput technologies to measure everything and see how these features, these molecules are connected, it includes, of course, the microbiome and the immune system, you are going to end up with something like this, hairball. Okay, so basically, you can do much without quantitative biology, without knowing graph theory, network analysis. So what we do in the lab is to try to make sense out of this hairball. Is try to reduce the amount of stuff that you need to analyze and try to see if this can be able to predict an outcome. Okay? So, and where do we get the data to create this hairball here? We can get directly from human studies or experiments from in vitro or animal models. We can also extract information from text using text mining and machine learning. And also things from not related to molecular biology or health, such as Twitter, Google Maps, etc. And what I want to say, maybe the most important message here is that what you guys are learning, bioinformatics, computational tool analysis, is only gonna serve to reduce the amount of features or network to look at. Okay, so it's a very important process, but these parts here, to interpret the data, you still have to be a scientist. Okay, so you cannot neglect your scientific background that you are studying in biology, ecology, whatever, and acknowledge that the tools are only tools, only serve for you to reduce. It's not going to give you the hypothesis that your thesis, okay? So your PhD thesis is not going to be, oh, my thesis is analyzing transcriptomics, blah, blah, blah. This is not your question. So we have to always find what is the question that you want to answer with the perfect tools. These are the publications of our lab showing that we have publications on systems vaccinology, machine learning, immunology, and here are some of the infectious diseases that we worked: herpes zoster, malaria, chikungunya, Zika, dengue, meningococcus. So all these diseases are relevant to Brazil. Ebola is not, but I hope it's still not going to be important. But these are the kind of work where we do, and I'm not going to go into the details of these papers because this is very specific. Of course, when the COVID-19 pandemic started, we shifted everyone to work with COVID-19. We participate in over 30 papers on this disease to try to understand what happens with the disease. But as you can see here, it was not only systems biology. We were also doing epidemiology, developing tools, even diagnostic methods. And how network medicine can be applied to precision medicine? Well, the traditional medicine is a descriptive medicine. You try to describe what the patient has. You try to do a diagnosis that sometimes is empirical. You don't know if it's going to work or not. And what precision medicine is trying to do is to understand the disease mechanism. And also, if you understand the disease mechanism, you can provide a mechanism-based diagnostic. And instead of grouping the disease by organ, you can group by molecular classification. And then you can deliver individualize treatment. You can prospectively treat someone, avoid the person to have the disease. And instead of treating the sick people, you try to talk to the health people to not become sick in the first place. So these three parts here, to understand the disease mechanism, you need the network medicine. You have to combine the information of clinical data, immunology, molecular data in order to identify the mechanism. And then you can apply this to individualized treatment, prospective evaluation of risk, etc. The network medicine, the jargon, was actually given by Barabasi, which is my idol. I love this guy. So a few years ago, he actually invited me to go to his lab in Boston to meet him, and as a big fan, I was like, this is the guy. And then I think he thought that this was a Brazilian thing, and then he also did this, but I was very proud to be beside him. So he is the guy that developed the term network medicine, which we use. So imagine you have two conditions, one healthy and one sick person. What you can do is to use the high throughput techniques to measure the features, and then you can and then you can combine, you can integrate this using network analysis. So you can use protein-protein interactions, text mining to combine the features. And this is the network construction part. When you compare two different networks, one from sick and one from health, that's the network medicine, okay? And why it's a different way of analyzing the disease? Because most people, when they are analyzing large amounts of data, for example, let's measure the activity of 20,000 genes in cancer and in normal tissue, okay? The first thing people usually do, especially in the back old days, was to focus on that gene that was changing the most. Oh, this changed 10 times, so that's the most important gene in cancer, for example. But do you agree that maybe this guy here, isolating the network, impacts less the biology than this guy that is connecting different modules, but is very central and it's changing less. It's only increasing 5 times. Or even this, can you agree that a lot of genes from the same modules changing a little bit may be more important biologically than one single guy alone in the periphery? So network medicine analyzes the whole network, the context of the gene in the networks to see if this can impact the biology, okay? And what do we do in our laboratory? So this is Bruna, your friend, Natalia. So Bruna was in the barbecue of the lab, and this is the computational systems biology laboratory. So what we do in Albert Einstein is to use systems biology for health conditions. The only project that I'm going to talk about, which is not on human disease, is spatial transcriptomics, because that's a new stuff that we are dealing, and I really enjoy doing this. And of course, we are now applying this to cancer, but I'm going to show some things that are related actually to the mouse brain, okay? We have data from 3 sections of the mouse brain, you see? And when you remove the section, you can measure the activity of hundreds of genes within each single cell spatially, okay? And if you put all the gray dots here, each gray dot is a cell, okay? If you overlay what people knows in the textbook about the brain anatomy, you're going to see that the textbook is going to fail to find out which region is this, because this is going to be empty, because this is a real sample. How can we know like which regions these parts here belong? If these parts, you know, how can we overlay what people know about the regions and the data that's real? If we use the overlap methods, we're going to fail. And how can we solve this? To solve this, we went to the stars. We went to astronomy. And we saw that the same tool that is used to analyze stars, instead of looking at stars, you can look at single cells. This is exactly the same thing. And then when we apply these methods that astronomers use, we can correct the shape and then find out which region is this. And then we identify which cell type belongs each one of these 700,000 single cells. And then which ones are astrocytes, endothelial, the term doesn't matter much. And of course, you have to do dimensional reduction. You probably already learned this, or you're going to learn, right, Flavia? Yes, you will. And then you can place back these cell types in the brain. So these are the three sections, and these are the different cell types. You see that some cell types are like randomly distributed, and some cell types are specifically located in parts of the brain. And that's really interesting. What is more interesting is that if you take, for example, two types, which is neurons, and then you cluster again, you can identify subclusters. So although these are inhibitory neurons, this is cluster 7, this is cluster 6, okay? And then you put back in the image. Look at how beautiful. You can see that although it's the same cell type, depends on the signature that the cell type has, they are located in the different parts of the brain. So an specialist on anatomy would say this is an inhibitory neuron, but actually this is an inhibitory neuron that is specifically located in this part here. They have a specific signature that only these neurons have. And that's something very interesting that you can only get from spatial transcriptomics. You cannot get from single cell transcriptomics. And then you can look at specific genes across all the tissues. You can also get one cell type, for example, and this is what we are doing now, and finding which cell type is activated, and then you look at what is its surrounding. What happens with the cell that is activated with the cells that are surrounding these cells? What is the impact of this? Can you guys think in an application of this for cancer, for example? Imagine like a microenvironment of a tumor which has not only the tumor cells, but the healthy cells, immune cells, everything like mixed there. And then you can see what happens with the CD8 T cells that is destroying the tumor cells, you know, and spatially. And that's, it's going to be the future. Instead of like biopsy using image, they are already trying to do spatial transcriptomics of cancer biopsies. Because then, in addition to the image, you also have the information what is activated, what is not, what is dying, what's not, okay? And of course, imagine now you have a table with 700,000 single cells, and within each single cell you have the XY location and also the expression activity of thousands of genes. And that's why you need the quantitative biology. Another analysis that we are doing is comparing two parts of the brain, the left and the right, which we were assuming that they have similar expression. But when we compare the same cell type, one from right, one from left, we see that there are differences, and the difference is actually interesting. So meaning that your brain works at molecular level very different if you're looking at the left or the right part. This is just an animation of to show like the two parts of the two genes, but I'm gonna shift to here. Because I'm a biologist, because I learned that we are generating more data than we can analyze. Every time that I start a project, I try to think on the biologists, people that do not know bioinformatics. So there is still a huge cliff separating people that generate the data from people that analyze the data, okay? So what our lab tries to do is to create a bridge that allows people that do not know how to analyze the data. So that's the girls from the lab, and these are the tools that I'm going to talk about. So I'm not going to focus on the data results, but the tools and how we can apply to biology, okay? And that's a good start, because instead of you already learn hardcore computational language, etc., you can start from what is already known, exists, and then you improve that tools, or you think in a different application. I'm not going to talk about all these projects, of course, but I'm going to talk about some of them. SemiTool is the first tool that we developed, and was developed by Pedro and then other people in the lab. And that's, it's because if you look at a network in biology, you're going to see that the genes that are in the same modules, the genes that are connected to each other, they tend to have similar co-expression patterns, okay? So if you measure the expression of these genes in green, you're going to see that they have similar expression patterns. When you increase the expression, the other one increases. When the gene decreases, the other gene decreases, okay? Why? Because they are regulated in the same way, right? With the same transcription factors, or one regulates each other. And same thing here. Our goal was to do the opposite. Let's start with the transcriptomics, and then let's identify the modules. So it's just analyze the transcriptomic, and then find what are the modules of genes that are co-expressed in that transcriptomic, okay? After you identify the modules, what you want is to see what the module is enriched for. What are the pathways that are belonging to these genes here, for example. And then you do the pathway enrichment analysis for each one of the modules. And then you want to integrate this with other type of data, for example, protein-protein interaction data, to see which receptor or which transcription factor is driving this transcription module here. SemiTool, the tool that we developed, does everything for you. So you only need the expression data, and then it's going to identify the module, it's going to do the enrichment analysis, and do the integration with the protein-protein interactions. We even created a web tool. You can play here, semitool.sysbio.tools. All the tools in our labs finish with sysbio.tools. And you can just drag your files, and then you're going to identify the modules for you, do the enrichment analysis. You can quickly see if your data is structured. You can use for transcriptomics, but also for any other data which is structured, such as proteomics, metabolomics, and maybe even ecological data. Because if you have a table where features could be species, whatever, in rows, and then the conditions are in columns, as long as you want to find correlations within the rows, the features, you can use this tool. But of course, it's not gonna work the enrichment analysis, because you cannot find out if there will be more ants or more palm trees. This is one result showing that if you run this in a Nature Immunology paper, you have different modules, and the modules are enriched for different stuff, and you have some hubs genes, which are nice genes for targeting for drugs, for example, if you want. These two here, MetaVolcano, it was developed by Cesar, and I'm going to show how we apply this. As you know, anyone can die of COVID-19, right? So, but we know that some people that have diabetes, people that have hypertension, people that have chronic obstructive pulmonary disease, people that have heart failure will have higher chance of dying of COVID-19, okay? Don't worry, because none of them here died, because none of them exist. They came from this website. This person does not exist. But that was the first question that we had, like, why completely different comorbidities increase the chance of someone dying of COVID-19? Because they are not the same disease. What are the molecular factors that are associated with severe COVID-19? We did this question even before the first case happens in Brazil. This is to show like an example. The World Health Organization declared a pandemic on March 11, and we submit the paper on March 27, meaning that less than 1 month we had the paper done. And how did we do this? We went to a data repository called GEO, which contains a lot of transcriptomic data, millions and millions of samples, and then we extract only the tissues, only the studies that were from human lungs of patients that have comorbidities that were associated with severe COVID-19. You understand? We only want to look at what people with different comorbidities have in common. And to do this analysis, we use the MetaVolcano. MetaVolcano is a tool that do meta-analysis from different transcriptomics. But you can also use any again, tables that have p-values and coefficients interval, etc., because it's going to do the meta-analysis for you. So when we do this, we find what are the genes that are consistently changing on these different comorbidities. And this is the Web 2 version, where again you can just drag your file, and then the tool is going to give you like a result like this, a meta-volcano, showing the meta-analysis, what are the genes that are consistently up or consistently down, or metabolites, or whatever you want to compare. And one of the findings here for the people that are not from biology was that one of the genes that was changing in most of these patients were ACE2. And ACE2 is important because it's the main receptor of the SARS-CoV-2, the virus that caused COVID-19 to enter the cell. So the hypothesis was that, oh, wait a minute, so maybe people that have different comorbidities have higher levels of the receptor in the lung. So when they are infected with the virus can easily find a cell to infect, and that's going to increase the SARS-CoV-2 replication, and that's going to increase the risk of developing severe COVID-19. So very simple hypothesis that we use public available data to show this. And then we show that that really happens in different comorbidities, and that came out in this paper that is cited in over almost 300 other papers. Molecular degree of perturbation. So who's controlling the time? You, Flavia?
Yeah, but you can go.
Okay, so okay, great. Am I speaking too fast or? Okay, cool. If you want to interrupt, you can also interrupt, okay? But molecular degree of perturbation, guys, that's started with Melissa and finished with Andrea. And that was the inspiration for this. Each column of this heat map is a different person, and each row is a different gene. And these are the people that do not have tuberculosis and do not have diabetes. And this is the people that have diabetes and do not have tuberculosis. These are the ones that have tuberculosis but not diabetes, and this has both diseases, tuberculosis and diabetes. Do you think that they look the same? No, right? Because you see that these guys here are very different than these guys here. These guys here are different than this one. And even the healthy people, these guys here are different than these ones here, right? You see, that's the first slide that I showed, because each human being is unique. So even if you have the same disease, your molecular profile is very different. And that's why precision medicine is important, because maybe the person has the same disease but also has other disease, or it's healthier in the spectrum of the disease. And that happens to every single human disease, the complex ones. And how can we quantify this heterogeneity? How can we quantify the perturbation of a sample compared to the non-perturbed samples, the healthy group? We create a very, very simple mathematically. Please, the mathematicians, do not mock me, because that's a very simple methods, but it works. What do? These are the control samples, the people that are healthy, and the rows are the expression of the genes. First thing we do is we calculate the median and the standard deviation of each gene, okay, for this group. And then we ask, for example, look only in this first column here, the first patient. So this gene here, the expression here, we compare the expression of this gene for this person with the standard deviation, the median of the healthy group. So basically, we count how many standard deviation is above or below the median of the health group. That's for one gene for one person. And then after we do this for everyone and all the genes, we transform this. These are the standard deviation. So these are the expression, and these are the standard deviation, number of standard deviation from the median. And then we just transform this into absolute value. So the negative is going to be positive. And then we sum this column. And that's going to be the score of this person compared to all the control persons. So taking into account all the genes, how far this person is from the healthy group. Okay, so that's the perturbation score. Very, very simple approach. But when we do this for the disease, we see that these are the people with diabetes, these are the people with tuberculosis, and these are the people with tuberculosis and diabetes. We thought that if you have two diseases, your perturbation is going to be higher, but actually it was not higher. It was not higher because diabetes has an epigenetic influence that shuts your transcription actually down. And if you look at each single person, which is these dots here, you're going to see the heterogeneity here. These people here looks almost healthy, you see, at molecular level. And these people here are very perturbed. Even the healthy people, you see that these three people here look like outliers compared to healthy people, because they may have fever, they may have eat rotten food and became sick, and that explains why they are not actually so healthy. And that influences your quantitative analysis. When you are doing a class comparison, when you are comparing one group versus another, if your group is too heterogeneous, that's going to affect your p-value, right? Because most of the methods are going to try to find the biggest difference between groups when you have low variation within the groups, right? So if you use this tool, you can actually compare different diseases, the perturbation of different diseases, but also identify outliers, also differentiate subgroups of people within the same disease, and then do the analysis again, only separating now the people with the same molecular perturbation. Okay, so that I'm not showing the paper, but give you more information about the disease, because you are dealing with a more homogeneous group. And we also have a web tool. You can just go to mdp.sysbio.tools and again drag your file, use the default parameters, and then run MDP, and it's going to give you the results. BioFits is a tool that we have not published yet, but this has to do with what I said. We are generating more data than we can analyze. And imagine now you have single cell RNA-seq. You have a group of people with the same disease, which is heterogeneous, and then you measure in the bloods the expression of 20,000 genes in 10,000 single cells for one person, and then you have hundreds of persons. So you are going to have a huge amount of information. And what people do is to create this computational methods that's going to reduce the stuff that you're going to look at. These are what I mentioned you. But even when you reduce, it's still too many, because you're dealing with 300 differentially expressed genes, 500 differentially expressed genes. How do you know which one of the genes you're going to use to create a knockout mice? You're going to do a functional experiment. You're going to develop a drug against that target, right? Even 100 genes is too much. So how can we reduce this even more? That's why we are trying to develop this BioFits tool, which look like this funnel, because it works actually to any biological data. You can input cytokines, proteomics, microRNAs. Each one of these is a different biological feature. And then the funnel, the BioFits, are going to try to identify what is the most important features in two groups that you are comparing. Okay? And it does so by using feature selection methods and machine learning algorithms. It kind of is an ensemble tool that gets your features and try to give you the most important ones. This is not the output of the tool, but it's just to show that when you start with 20,000 genes, instead of looking at 300 genes, you can look at these 14 genes that is the most important in predicting two classes. And then from these 14 genes, you can read papers for each one of the genes, and then go back to the bigger list and see if it makes sense, and then you start designing your functional experiments. So that's the goal of the BioFits. Well, knowledge networks. This one is because of a situation like this. Imagine that you have an intellectual person that wants to know more about a drug. Okay, so the person will go to the literature and try to find, okay, how do I learn more about the molecular mechanism of chloroquine treatment against COVID-19? Because that's something that person do. Probably this intellectual person went to PubMed, typed chloroquine in PubMed, and then he found a paper like this. The first thing you do when you read the title, you see, oh, chloroquine suppress this gene called NLRP3, NLRP3, okay? So the person goes in the notebook and writes, chloroquine inhibits NLRP3. And then the person reads the abstract, and then they see chloroquine prohibits expression of IL-1 beta and IL-18. Prohibits expression is the same as inhibits, right? And then the person draws this inhibits, okay? And then the person says, okay, this paper is read, I can move on to the next paper. But then when you go to PubMed, you're going to see that only chloroquine have over 25,000 articles. So one person cannot read all the papers from one single drug, okay? Well, that has to do with what we are all facing, which is this. Since the World War II, we have been publishing in the year 2000 500,000 papers per year only in PubMed, okay, which are the biomedical literature. In 2019, we published 1.2 million. In 2020 and 2021, 1.5 million papers per year. So it doesn't matter your area of expertise, you can work with beetles of these species, you're going to see that there are more papers that you can read. We are all facing this knowledge tsunami, okay? So we cannot simply read all the papers, even from your subfield. And well, you cannot read the 30 million papers that exist in PubMed, but the machine can do this, right? That's what we did. We used artificial intelligence to read all these millions of papers automatically, and then identify the relationships between genes, drugs, and disease, create this knowledge network, and then with this knowledge network, we human beings can interrogate and do the network analysis, etc., to identify which drugs or genes or disease are important for different contexts. And that's what we did as a proof of concept in 2020 for psychiatric and neurological disorders. We took all the papers from these diseases and create this knowledge network, and then we could see which are the factors that were in common to different diseases, very different diseases. So Alzheimer's, Huntington, we look at which ones are related to glutamate, which ones are related to inflammatory response, etc. And that was interesting for basic science. But we can also use this for repositioning drugs, which is you take the drug that was developed to treat, for example, Huntington disease. And then because you have the knowledge network, you see that several of the genes are shared between Huntington and Alzheimer. And then you can say, I'm going to repositioning this drug to treat a different disease, okay? And if we do this, we did for all the psychiatric and neurological disorder, and we found several drugs and antibodies that if you go to PubMed, you're not going to see a paper saying that, oh, you can use this drug to treat Alzheimer. But if you go to Google or to clinicaltrials.gov, you're going to see that some of these drugs are actually being tested if they can treat Alzheimer. That validates our methods, showing that the big pharma are not going to publish a paper giving you the idea of treating the disease, but they have to describe this in clinicaltrials.gov, for example. And we use this information to validate that this approach can give you potential candidates for repositioning drugs. And we also published this paper where we did this approach for almost 100 human diseases. And we learned that our knowledge increase differently if you are dealing with infectious disease. Guys, you cannot read, right? Or it's just me? Okay. Well, this image I generate this morning. So if you follow me on Instagram or LinkedIn or whatever, you're gonna see this, which is basically all the research on monkeypox that we use machine learning to read 700 articles, identify the embeddings, the how a machine would interpret each one of these 700 papers, and then compare these models of natural language process to see which article is more similar to each other. Okay, so that's how we create this figure here. And then we saw that this group of papers were dealing with the treatment of the disease. This group of paper was dealing with the fact that monkeypox is more relevant if you are pregnant than if you are a healthy adult, etc. And then we organize the literature using machine learning. We also create this animation that shows the number of papers published per year for different disease. So in 1947, most of the papers were published related to peptic ulcer. And that's what happens over the years. This is tuberculosis, showing that tuberculosis was very much studied in that time. Then it became hypertension, infarction for a while. And of course, you guys remember what happens in 1985? AIDS. So AIDS appears here, then the HIV, which caused AIDS. And then in the year 2000, obesity appears as a disease very much studied. If you want to cheer for the disease, you can go diabetes. Yeah, look at what happened in 2020. Guess which disease is this? See, like everyone stopped what they're doing to work on COVID-19, became the COVID expert, etc. And we saw this reflects on the literature. And because we have analyzed 30 million papers, we also create these other websites. Oh, by the way, guys, if you go to this website here, sorry. It's going to appear here. Sci70.sysbio.tools. You can do your own animation. You can choose which disease you want. You can choose the speeds. You can choose how many bars you want to do the animation. And then it's going to animate for you. You can put in your lecture, whatever seminar you want. Okay, and then you can reproduce this. We also create this other website called knowledgenetwork.sysbio.tools, which give you the research trends, how much people learn about over the years of any topic in biomedical fields. So, for example, you can type here obesity, for example, and you can type there HIV. And then you can change the parameters or not. You can change the speeds and then you do play, and you see that this is the research trends. How much people were researching on obesity or HIV. Of course, nobody knew about HIV until 85, and then you can learn like how they were learning about this. And then you can compare two different topics and put in your lecture or seminar. If you put here research articles, that shows the absolute number of papers, not a trend anymore. And then you're going to see the absolute number of papers per topic. Okay? If you go to our website, csbiology.org, which is the lab websites, you can find most of these things here. Some of you're not going to find because I do not update frequently the website. We also create this other tool. It's the same initial, but then it's countries, because then what we want to know is how much each country research on different topics. So, for example, did you know that leishmania is one of the most studied subjects in the world? Brazil is a country that studies leishmania more than any other country in the world. And this is the total number of publications, but then you can normalize by the total publication of the country. Otherwise, the US is always gonna be the top one. And then when you do this, you're gonna see that some interesting research that is being doing for different research. For example, microRNA is something that is studied a lot in Iran, for example. I did not know that. Curious, guys. This is something also very brand new that we are working. And do you guys know, of course, the Alan Turing test, right? The Alan Turing test, in a nutshell, tries to test if a human being can tell if you are talking or typing with a computer or with a human, okay? That's the test. What we did was to create a tool, the Alan Turing test for scientists. We use machine learning to create texts from an abstract, for example, or to create text for you. Let's say, for example, what we did, we took 1,000 nature papers and science papers, real nature and science papers, and then get the abstracts, and then use the abstracts to the machine learning to propose a title, only based on the abstracts, okay? Imagine that I give you an abstract and you have to propose a title just based on the abstracts. We also, of course, use the original title, and that's the one that was written by the original authors, and we use queries to generate the title and then put it here. And that's you. You have to tell if the title that you are reading is actually written by an AI or written by a human being. And that's where the tool is. Do you guys, nobody has, can you guys try? It's 10 seconds. There is no login, no nothing. You just go here, crowdsnotation.tools. So after you play this game, you tell me what was your score. I know that you're going to do very quickly. You're not going to read the whole abstract, everything, but the abstract is going to be there. Crowdsnotation.tools. Bate às 4? Então beleza. Okay, guys, did you guys finish? Okay. You don't have to read all the abstracts, but I do want to know if you are a physicist, for example, and then you find an article of physics, then I want you to really pay attention. If you are a biologist working with the subject that you are reading, then I really want to try to see if you can tell if it's a machine or not. Or of course, if you are a biologist and then you see a physics paper, you can just guess, okay? But try to pay attention if you got it right or wrong in the subject of your expertise, because that's more fair comparison. Did you finish?
4 out of 10.
4? Yeah. Come on, try again. You can do better. 4 is even worse than the random stuff. Like you can just like do it like this. Come on, guys. 5 minimum. Seven?
I got 6.
6. 6 is most of people do 6. I also got 6. Who got 8? You got 8? Wow. Does anyone got 9 or 10? Okay, everyone finish? 2? Wow, man, we got a winner here. Yeah, after a while, if you start playing much, you're going to start seeing some patterns. You're going to start seeing that, oh, if the title looks too beautiful, that's probably a machine. If there is a dot in the end, it's human. Some bugs. That was actually a bug that we didn't see. but one of my PhD students found that. Did you get 8? Nice. But you did together or? Ah, come on. Okay, so did you guys find any paper that was from your field? And did you get right or wrong? Did you get right or wrong? So that's the kind of research that we had to decide in making like a tool that people will try fast. Because if we do a login, asking people what is your expertise, because that was the ideal. Like, let's find out what is the expertise of the person and then see if the experts will get it right more frequently than non-experts. But then we decided to do it more and fast. So if we do this, we start seeing like who got it right and who got it wrong. And I don't know the numbers now, but apparently most of people were actually finding like a score of 6. And what we are going to do now is try to analyze this, because we also measure the time that person spent on each page. So we can see if the person that spent a lot of time, we will actually get more correct answers than. But the best would be to see if the experts get it right more. But the most important part here, guys, is for you to acknowledge that the machine actually can give you a title that is the same quality of a Nature and Science paper. You don't even need to come up with your title anymore. You just go here and generate for you. Actually, if you put, for example, hepatitis C, it writes already a nice paragraph for you to put in your introduction. Yeah, it's like very like scary actually, and because it gets a lot of things right. And that's what we use for the monkeypox also research, because the title generation is only one part of the tool. Well, systems immunology and inflammatory disease projects. That's a little bit more biology, but just to tell you that hundreds of millions of people have the same kind of chronic inflammatory disease. And it's a heterogeneous group of disease. Okay, so what we are doing, and I put this project here because this is nice repository for you guys to play in your course, which is the data that we use that is already available in the data repository. So what we did was to create this project called Systems Immunology of Inflammatory Disease, where we collect hundreds of different disease studies, organize them, and then try to analyze, integrate them to understand what is common and what is not between very different inflammatory disease. And that's a repository where you can play with a lot of data. We have data from more than 30,000 human samples. And what we are doing now is to try to merge these different networks here. And for example, the point that we are now is try to see which tissue is different, because you have inflammatory disease, but you have tissues which are blood, lungs, synovial tissue, joints, everything. And then you can see what are the similarities between tissues and diseases at molecular level. And we are also doing this for COVID-19. And Transcriptomic Atlas is important because if you go to the literature, there are over 50 papers on transcriptomic studies of COVID-19. But the problem is that if you take 2 papers randomly and then look at the genes that are differentially expressed, reported by the authors, you're going to see that the overlap between them are minimum. So there is a little consistency between different tissues. So every author says, oh, these genes are the ones that change in COVID. No, these genes are the ones that change in COVID. And when you're going to see, it's not the same. You know, how are you going to trust what are the genes that are really affected by the disease? And that's what we are trying to do here, to create an atlas by combining these different studies. So for example, if you take the blood tissue, we can see that there are like 8 or 9 studies on blood. And if you look at the number of differentially expressed genes that appears in at least one of these 8 studies, you're going to see that there are over 5,000 genes. what can you do with a list of 5,000 genes, right? But if you want the genes that is differentially expressed in all 8 studies, you're going to find 0 genes. So what we are doing now is to find a compromise here, try to see like what are the genes that are affected in most of the studies, because this is more reliable signature of the disease in the blood. And then we do this, for example, for the blood, and then find 206 genes. And then we apply the same approach for liver, kidney, colon, heart, nasopharynx, everything to create this atlas. And now what we are doing is comparing these different tissues to see like what are the genes that appear, that are affected by COVID in all tissues measured. And then these are the main network, the hairball network, with a lot of genes in common and some genes that are specific to the tissue. And of course, we're going to create a website where you guys do not need to redo the analysis, but just consult, just search for the gene that you want, the tissue that you want, and then you find if the tissue or the gene is affected. PEBA is a tool that we have not published yet. And is the name of a species of armadillo. But the reason of the name is different, but I'll tell you. Imagine that you have an expression table where you have the columns are the samples with healthy people or malaria, and the rows are the genes. This is usually a transcriptomic file, okay? 20,000 rows and hundreds of columns, which are the samples. The first thing people do is to do a differential expression analysis, which is get for each row, compare the values of one group versus the values of the other group for that single row. And then you do this for all the rows. And then you have 25,000 p-values, 25,000 log2 fold change for every single gene. And then people use this list of p-values and fold chains, define a cutoff. Let's say adjusted p-value of 0.05 and log2 fold change of 1, which is fold change of 2, to say that these are the differentially expressed genes. And then you get, for example, 200 genes. And then you do the pathway enrichment analysis, because I want to know what these 200 genes do, like what are the pathways that are enriched. And this is the functional genomics. When you do this, you identify the biology that is changing in your condition, in this case, malaria, for example. But there is no statistician that obligates you to use an adjusted p-value of 0.05, right? You can use adjusted p-value of 0.01 or 0.1. You can use a fold change of 4. You can use a fold change of 1.5. There is no one single answer that you have to use that cutoff. And if you change the cutoff, you'll find, for example, 500 differentially expressed genes, and then you do the enrichment analysis, you find a pathway enriched list, which is different than the one that you have with 200 genes. Do you guys agree that this is very arbitrary, the cutoff that you use? Because if you change the cutoff, you're going to change your results. And that's when I explained this to the problem to the first out of the paper, he was from Bahia. And then he said, oh, I think this is very PEBA. For the Brazilians, some will understand that PEBA is like a bad thing. I learned from him that PEBA is like, oh, this is bullshit. And that became the name of the tool. Because it is indeed very PEBA, right? An arbitrary cutoff. So what the tool does, it's a web tool that I think it's already online, but you can just upload your table. And this is not an animation, but what the tool does is a very simple thing. Like it's rank the genes based on p-values and fold change, and then get the top 100 genes or top 50 genes and do the enrichment analysis. And then they get the top 100 and do the enrichment analysis. And then they want top 150. You see, the tool only changes the cutoff, and then do the same analysis. And here in the columns are the different pathways, and in the rows are the different cutoffs. These are the top 100, top 200, top 300, and these are the different pathways. So this is just for the user to see what are the pathways that only appears enriched when you use a specific cutoff, and what are the pathways that appears regardless of the cutoff. So if you want to do validation, do you agree that I would prefer to write a thesis on the pathway that is independent of the cutoff instead of putting all my chips on a pathway that only appears in a specific cutoff? So the tool only shows you, visualize the results for you to decide which cutoff or which pathway you're going to focus on your research. It's almost over, guys. Digital epidemiology. What did you guys try to learn during the COVID-19 pandemic? Korean? French cuisine? Come on. I tried to learn ukulele. I failed. But tell me one thing that you tried to learn in the beginning. Japanese. Did you fail or did you finish? Wow. Cool. Does anyone want to share your... What? Oh, really? Cool. And did you succeed or? That's awesome. Yeah. So if you use Google search, which is called Google Trends, you can actually find out what people were trying to learn. And Korean was one of the languages that people were trying to learn. Guitar, some cooking, you know, like making breads. These are things that people try to learn. So that's why I kind of know like what you guys probably did, because statistically you can find what people are doing. And also you can use this to find what are the fears of people, what are the curiosity of people, like what are the emotions of people during the pandemic. And then you can correlate this to see if this can predict what's going to happen with the cases of COVID-19, for example. And this is what we did. We looked, for example, in São Paulo, when people start, we look for several different keywords, what people were typing, what people were searching. And then we saw that when people were when searching for COVID, a few weeks later, the number of cases increased. Then people stopped searching, the number of cases decreased. People increased searching, increased cases. Decrease, decrease, increase, increase. You see, there is a correlation in São Paulo. If you do the same thing for South Carolina in US, it also correlates really, really well. If people search, increase the case. If you stop searching, the case decrease. If you do this for this region of India, this also correlates well. In fact, if you do the same thing for all the states, for almost all the countries, you're going to see that there is a beautiful correlation, meaning that you can use Google search as an epidemiological tool, very, very cheap, to monitor when the cases are going to increase or decrease, instead of like doing the RT-PCR anymore. Tukuchi is a tool where I did because we are looking at the 40,000 UBS, the Unified Health System, the primary care facilities of our SUS. And the problem with this, guys, is that we have different databases for different diseases. We have one database for malaria, one for syphilis, one for HIV, and these databases do not communicate to each other. And that's a problem, because sometimes you want to find out what is the person that is here in this database and is also here, right? So let's take an example of Luis Mendonça. Luis Mendonça was born in October 6, 1988, and his mother is Teresa. What happens is that Luis got sick, they went to a hospital, and this information went to the database that was sent to the governments. And the same Luis was vaccinated a few months later, and that information went to a database and also went to the governments. And frequently, we scientists want to know if Luis that was vaccinated already had the disease, or if Luis have the disease A, also have the disease B. And that's what we need to do with record linkage, which is linking these different databases using the same records. And what is the records? The records is the personal information. If you have the CPF, a number, it's easy to link. But most of the time, the database actually has this information. And the problem is that you're going to see here that Luis with Z, Luis with S, empty spots, a number. So how can you do record linkage if there are so many typos, so many grammar errors, you know, like misspelling? what was our idea? Our idea was to transform the personal information into DNA sequence. So let's convert this information into DNA sequence and then use a tool, repositioning a tool that already deals with genomic information, knows how to do with mutations, etc., which is BLAST. So we use BLAST to compare the sequence and see if Luis was present in both databases. So we did not have to reinvent the wheel to create a fancy record linkage tool, because BLAST is a very fast and efficient tool. The only trick here was to convert the personal information into DNA. And then when we do this, using this generic wheel here, we can convert each single letter into codon, and then we compare the sequence with BLAST. For example, we can also change the way we do this conversion. For example, the M from Maria can be GGT, but if we change here, it could be also TCA. So if you find a DNA sequence, it's hard for you to decode what is the person that is there if you do not know how to the wheel was structured, okay? But this, of course, this encryption is not very powerful, because if you have millions of sequences, you can definitely, knowing the frequency of names, etc., find out what is the... But for a few sequences, it's very efficient. And then what Tukuchi BLAST does is to convert this information to sequence and then compare. But then it becomes the problem of the cutoff again. How do I know if the similarity is enough to say that Luis is in both databases, right? Because this is a probabilistic method. And what we did was to use machine learning on real data sets to define the parameters and cutoff that are going to be used to say that these are the records that should be linked. Let's say, for example, let's use Fernando Bosa, which is a friend of mine, Fiocruz. And this is how Fernando Bosa is translated into DNA. If you write Fernando Bossa, this is going to be the Fernando Bossa in DNA. You see that when you run BLAST, you're going to see that only 2 letters are actually different between these 2 records. But this is the same person. But how do I know if this score is enough to say that this is the same person? If we take his brother, which is also a friend of mine, Marcelo Bosa, this is going to be his name in DNA. And then if you try to run BLAST, it's not going to be significant, showing that these are different persons. But how can I really compare? Let's put the name of the mother of Fernando Bosa and Marcelo Bosa, which I don't know the name, but I'm going to put Fernando Bosa, mãe do Fernando Bosa. And then we do the same for Marcelo Bosa, which has the same mother. And that's mãe do Bosa here. Then when you run this, you're going to find that there is a perfect match. But perfect match because they start from different regions. BLAST was ignoring this part here, okay? So what the machine learning model do is to use the parameters, not only the alignment scores, but also the coverage, like what is the sequence that the entirety of the sequence or not. And then we define the cutoffs. It works well for Portuguese, but of course, if we're going to do it for database in English, we're going to have to redo this in a real database in English. We also create the tukuchitail.csbiology.org, which allows you to do the gold standards. How do you evaluate the accuracy of your methods? You have to have a human to go there and say, like, okay, this is Fernando Bosa, this is Fernando Bosa, this is not the Fernando Bosa. And then we create this tool that allows you to quickly, using the keyboards, the left and right keys, to say that if it's the same record or not. And that allows us to do this for thousands and thousands of records to have the gold standard to compare our data. When we do this, we found what are the most frequently errors. For example, M and N was very frequently mistyped. And because if you go to the keyboard, they are very close to each other. But why E and A, E and I? So we identified the reasons why there were so many errors on specific letters and numbers. We also use this to remove the redundancy of database, and then we create a database of 300 million Brazilians that do not exist to really stress out the system to see if it's going to work with a lot of records. And all the tools that we test, none of them worked. Only one worked, which is the CDAX RLL. And this is the time when CDAX was taking days to run, we could run this in a few hours. When CDAX needs a very strong computer, we can do this with a laptop. So this is a very fast and light tool. And we did this for real data sets, real database. Traditional records found that 84 people that die of this different disease also die in the central mortality database of the governments. Here you have accidents, you have everything. And when we use our methods, we found almost 200. Meaning that a lot of people were considered alive when they are actually dead. We also create this Tukuchi Translator, which allows you to type whatever you want in DNA. So if you want to send a message to your loved one in DNA, you go here, type your whatever you want, and then you send to the person, and the person is going to be, I don't know, happy or... Well, and why we are focusing on this, guys? Because we are living in a moment where the memory, the way we store memory in hard drives is going to be obsolete because we are generating too much data, right? Like if you see how much video you post online and photos, a single person do this. So we are reaching the point that we are not going to have hard drives to store all this information. But the DNA is a molecule that allows you to compact a lot of information, and it replicates, and it's very, very light. So you can put 2 terabytes of information in 0.0009 grams of DNA. And this is going to be also the future for biotechnology. So you're going to store your files into DNA, put in your freezer, and whenever you want, you retrieve the information. For example, they took the Shakespeare sonnets, and they convert this into DNA, put in a bacteria, send to another country. The other country sequence the DNA, and then convert back to the Shakespeare text. They did this even with a video, showing that you can reproduce a video by sending the information through DNA. And there are like companies already starting up to master this information, because the problem is you can put all your photo, MP3, whatever you want in a DNA, but how you're going to extract only the information that you want? You have to sequence the whole thing, right? So there are like a lot of challenges, but that's going to be very exciting. And if you are interested in this, I put money on this because I think this is going to be the future. Outbreak I'm going to skip because it's just a tool that we developed that shows that do the animation of cases for you. The ecology people can use this because if you have GPS information, you can just drag your, you can go to this website, put a title, a description, get the cases or the animals and the GPS location, and the dates where you have the person, then you upload here. It's going to show a map with a calendar where you put here to make an animation. So, for example, this is 700 million cases of COVID-19 COVIDs. So the case started in China, the orange is the case, and red is the deaths. And this is Europe. You can zoom in, zoom out, go to the location that you want. And that's very customized. So any GPS data that you guys have, you can put it in this tool and do the animation automatically. 350? Okay, guys. So I have to finish, but we have this project which is the government paid 8 million reais for us to develop an app, a cell phone app, that automatically reads the vaccination cards because they want to know where the people are protected or not. And that's not a simple thing to do, because we have to detect what are the vaccines that the person received, and then if the person received or not was vaccinated or not. CIPOS is a tool that we use the Google history to identify where the person went to see what are the hotspots of transmission. So we did this for malaria. We found where the people with malaria got infected. This is to show that this is before the pandemics. This is showing one person how much the person moves, and then it's going to become an app, so you can see, like, I'm going to go to this restaurant. Oh, no, it's a chikungunya hotspot. I'm not going to go there. But I don't know if you're going to be sued. Probably you're going to be sued. So tuberculosis, this is also interesting because we can get the Google history data of two persons, one with tuberculosis and the contacts of the person, and quantify how much exposure the person has to see if the person, what is the time that you need to become infected. And this is to people, a pair of people showing that they slept together. You see exactly when and how much and where. Of course, this is all ethics committee approval, and we do not share the information. We also do this for chikungunya. We can see that the mobility changes when you get sick. We did this for COVID-19. This is one person with COVID that was taking the subway. So when the person was infected, we see that this person take the subway because these are the subway stations. So guess how many people can get infected even before you get tested and know that you have COVID-19. I can see some people like putting the mask again. Yes, it's a very difficult disease to work with. And then we found the isolation score of people, but then I'm going to the last. So this is the Amazon Alexa project, and Thiago did this. So Alexa works for like personal stuff, right? Alexa, it's gonna rain, play a song, but then we decided to do this for the lab. So you can use, for example, the lab family. The mother is the one that knows where everything is, right? So you ask Alexa, science mom, where is the drug X54T? And then Alexa is gonna say, it's located in freezer 3, shelf 2. And it does this because it integrates with the Google Sheets. So if you put your inventory there, Alexa is going to tell you where everything is. The grandma is the one that do the recipes, right? So if you ask, like, read the protocol for me, it's going to give you the protocol. The grandpa is the one that sits in the couch and read the papers. So if you say, like, tell me about dengue, it's going to go to PubMed and start telling the papers on dengue. And everything's already in Amazon, so if you have Alexa, you can download this. And we even do the 3D printer support for the Alexa, so you can put in your lab without interfering with your equipments. So as a conclusion, I'm going to say that we have to make it strong the network science, not only the network analysis, but we need this collaboration. You cannot be the single person that knows how to do everything. And COVID-19 pandemic show us this. And that's why it's important for you to do not try to embrace the world and work together with other people. This is to show that we can do low budget science, but of course only for the analysis, but because all the money was invested already in training people, storing data, generating the data. And that's the team, and which I would like to acknowledge. And of course, thank you guys for listening to me 1 hour and 30 minutes. Quer acender a luz, não? Esse aqui. Tá bom. Você vai precisar disso?
Hi. Firstly, the code of those tools you developed, is it open?
Is it open?
Yes.
Yes, all of them are actually open because we use open source tools. There is a license when you use a library that someone else did. You have to make your tool available. So everything is either in the websites or it's in GitHub of the lab. Nice. So you can download everything, even the Tukuchi blast, everything.
Yeah, and about the Tukuchi, I found it very, very interesting that you decided to use it, to use blast in that, I don't know, I'll say it in the best of ways, in that perverted, deviant way that is to convert an AI. That's simple, but I couldn't imagine that coming up. So can you tell a bit more about the story of that?
How did I get the idea?
Yeah, and how that came about? Because
Yes, it was actually Bill and Melinda Gates Foundation grants. They were asked. Fiocruz has a building to do this record linkage. So they construct a building, they put a supercomputer there, which only 4 people can go inside. There's no internet connection. You know, the only way that the government gave this data to this building of Fiocruz was that this should be like secure, and nobody can access, and that's going to be a problem. And then these people start a Bill and Melinda Gates Foundation call for projects to do the integration. And most of the people were asking for analysis. Oh, I'm going to integrate this question, this information with this to see if Bolsa Familia impacts the health. You know, people were proposing the results of the analysis. I was actually concerned because when they put a call, they said like, okay, you can play with the data, we can provide you, but you have to go to Bahia, you have to have a supercomputer, you have to have 100 gigabytes of memory to run the analysis. I was like, man, I cannot do this. And then I was wondering like, which tool usually deals with a large amount of information. And then I thought genomic tools, because 3.2 billion base pairs, millions of reads. If you do BLAST, you can compare one sequence with millions of other sequences, right? I was like, oh, BLAST is the tool that is going to make the process easier. And then we proposed the grants because of this. So it was not like romantic or anything. I was surfing and then, oh man. So it was purely because of money. We did not get the grants, but then we did it anyways. So that's how we invented the BLAST Tukuchi. The beginning was actually, I didn't show you, but the original idea was to use GWAS. Do you guys know what is GWAS analysis? So for example, you want to compare what are the mutations, the genetic variations of people with Alzheimer and people that do not have Alzheimer. And then you collect the genome of 20,000 people with Alzheimer and 20,000 people without Alzheimer, and then try to see what are the bases. Oh, everyone here has a T, whereas everyone here has an A. So meaning that this T in this place is associated with Alzheimer, right? So for you to analyze this large amount of information, to do genome-wide analysis, you need a powerful tool. And that's my original idea, was to use GWAS tools to analyze, instead of each base is going to be like a base, each base could be like a feature. So this person has HIV, this person has tuberculosis, and then we can just use the same tools to quickly compare features, instead of like doing the record linkage. The record linkage came after we saw like, well, why not doing the linkage as well? So it's because our lab has people that have completely different background that allow us to do this.
Okay, first I need to thank you for this amazing seminar that was really amazing. And so I have 2 questions, and one is mostly practical. So I don't know how is the process to library prepare of the spatial transcriptomics, but it would be possible if we did this like to the microbiome of the gut and related the presence, because like we know that there is some correlation between a microbiome of the goats and carcinoma. We could relation this like where is this kind of disease and the transcriptional profile of the microbiome.
Yes, in theory, yes. In reality, it's not that simple, and because every single technique that I showed you guys, you have different phases. The first phase is the one that people are like establishing the technology and then is maturing. You know, like the transcriptomics, the RNA-seq now that you know now is after like 15 years of research, which now is a mature technique. So spatial transcriptomics, people have already, so usually what happens is that different brands start competing. Same thing with the tools and package, several things, oh, I have the best methods, and then after a while we see what is the winner, what is the ones that are really gonna fit. So we are now in the phase where we have very different techniques comparing which one is the best one. Some techniques that they claim is spatial transcriptomics, they are not actually single cell. They measure the transcription of an area, not the single cell. The one that I showed is a single cell technique, but the technique that's used, which is fluorescent immuno in situ hybridization, only allows you to do this for a few hundreds of genes. So you have always like a trade-off. Either you have a single cell spatially resolved, but only few genes, or you have a lot of genes, but not single cell. And we do not have yet the perfect, for example, methods that allows you to have single cell, a lot of genes, and also a lot of depths, which is coverage. Because once you have a lot of coverage, then you can do a mutation analysis to see like how the because one of the problems with cancer is that people get resistant to the treatment. So you treat the person gets well, but then the tumor cells that have mutations that are not sensitive to the treatment becomes the ones that kill the person. So if you know what are all the mutations that the person has, even spatially, that you can choose the drugs that you're going to treat. So what I can say to you is that very soon I think that you can design this kind of experiments. But nowadays it's very expensive. The ones that Brazil can do, which is using the 10x technology, is only area. And I don't think can give you much information about like that. But that's why we need to actually train people on the analysis first. And when the technology arrives, we can do this.
And my second question is like, where did this inspiration to create tools comes from? Because obviously, like every work in science is important, but when you are creating tools to improve the community knowledge, I think that is really important thing. So where this comes from and why all this is open?
That's a beautiful question, and that's something that I really want you guys to do the same. Like it's not that I'm brilliant. What I'm good at actually is to listen. If I sit down and say like, oh, I'm gonna come up with a tool, it's not good, nothing's gonna come up. But if you really talk to people, so if you go for coffee with your colleagues, even here, guys, if one person starts telling your problem to the other person, and so I'm good at listening like the problems, and then say, oh, maybe if I try to do this, and that's how actually I learned bioinformatics. So for example, the 8 million reais projects that the government paid, that was actually talking to my wife, which is a pediatrician, and she was saying that in the hospital that she works, the nurse takes a long time to get the vaccination cards of a kid, of a 2-year-old baby, and then the person has to see which vaccine the baby received, and then go to another table that tells which vaccine the baby should have received, which is not here, and that takes like 15 minutes, and then I was like, oh, so why not automatically detect the vaccines and also compare already in the database which ones is missing, and then it's gonna show to the nurse, okay, you have to put hepatitis B here in this kid. So it was a practical solution. And then I was in Manaus having lunch with a guy from the government that I did not even know that was from the government. And then I was telling him about this idea. I was like, oh yeah, my wife, blah, blah. Only this. And then he went to Brasilia, talked internally, and then one day he emailed me and said like, oh, we want to support your project. I'm like, what? Because there was this Manaus measles outbreak, people want to know what was the coverage of measles vaccination. And then I was like, okay. And so that's how, like, just talk too much. Like you have to. do a lot of conversations and listen to people and get inspiration. And what happened with the Tukuchi blast, for example, that one was, I saw the grants, the call. I asked my whole lab, say like, okay guys, I want to brainstorm to come up with ideas for this grant. I want this grant. And then we were like talking, and then I have the idea of the DNA stuff, and then 80% of the lab said, it's not gonna work. It's gonna fail because already SQL can do this, blah, blah. And even so, like there was one guy, DNA, that say like, oh, I wanna try this. So I have some crazy ideas. I have students that buys these crazy ideas, and then they go further. So if you look at innovation, there is this called TRL, Technology Readiness Level. So it goes from 1 to 9. 1 is when you have the idea, and then 2 and 3 is when you have proof of concept. And then 9 is when the product is already being sold everywhere, you know, like, so you have marketing, a lot of people. I like to be in the first 2 TRL. I want to have the ideas, and then I want someone else to make this as a product, to start a startup company, because I don't want this marketing part, this lawyer patents, and that's why all the tools are open, because I don't want to deal with registration.
Well, it's a more methodological question. We saw that different tools employs the network analysis to evaluate the relationship between these variables, such as genes, tissues, cells, and transcripts. I'm interested in know what's the method to evaluate these strengths of relationship between these variables, and if this method is common between these tools.
Good question, and I have not told you what are the network analysis parts here, because each tool has a different principle. For example, what do you prefer? Have 5,000 friends in Facebook to be like the hub of 5,000 nodes, or to have only 200 friends, but the 200 friends are Anita, Lula, all the important people, right? So same thing with network analysis. Some tools we want to find the hubs, to want to find what are the most important one. The other ones we want to find what are the ones with the highest betweenness, centrality, you know, like, so the way you identify the modules, the way you cut off the edge, this is gonna change according to the needs of the projects. I know that I gave you a vague answer, but just to tell you that we try all possible methods. We actually did not even invent any network algorithm. We use like a algorithm that's iGraph in R. It already give you like Python, already have like everything. So we did not reinvent any fancy network analysis tool. Was just the purpose. So if you go in the mathematics part here, it's very simple, guys. There is no fancy stuff. We do not even, because I'm more interested in solving the problem, biological problem, than to come up with a real fundamental methods that's gonna analyze in a completely different way.
About the
but with the correlation, we use like simple Pearson correlation, serum correlation to measure the co-expression. We do for integration partial least square regression, partial correlation, some like stuff that already exists in tools, you know. So I can list some of the methods that we use to find this connection, or we use experiments that were from the bench, that people say that the gene A is connected to gene B, the protein-protein interaction experiment. So it's just a matter of like selecting the database, which already have the edges, and then put into a network.
About the text models that you showed, do you think there are like important ethical concerns about the development of these tools? Because I think it actually goes both ways. If you have like artificial intelligence analyzing the papers you write, I think that that could influence how you want to write it, because you know that that thing is going to be mined for data, is one way this thing can influence how you write. But then there's the other way, which is like if you have artificial intelligence that can write things for you meaningfully, I think that could help, it could facilitate like forgeries and unethical behavior.
Do you guys are doing happy hour here? Because that's the question that I would love to discuss over a happy hour, because I do agree with you, like it's very scary, because if machines can start reading papers and also writing papers, where the humans are going to be, right? Because you don't even need the intermediates anymore. And that's true. Like, and one example that we are using was the Nobel Prize winner for identify the Hepatitis C virus. Okay, I took all the papers that the guy published on Hepatitis C, and then look at the introduction, the first paragraph of his paper. It's almost the same thing. How many different ways you can talk about the problem of hepatitis C, right? But because of self-plagiarism, you cannot use the same sentence. But what is the problem of using the same perfect sentence that you already wrote for a paper reproduced again, right? Like, so there is like, for one side, an ethical problem, because you cannot plagiarize people, but also sometimes, why not? Like, why not, how you have to waste your time thinking a different way of saying a sentence which already was perfect in the last paper that you published, right? And that's the thing that I would like to discuss and see what is ethical or not, because at the end, what does really matter? Like, is the sentence there, like, to give the credits for the person? You know, so it's I don't know like the answer for this, but I'm pretty sure that they're going to have to reevaluate this, because progress is not going to stop. These tools are already available for free. People can generate text, and all these texts that are generated are not plagiar, because if you try to see plagiarism, there is nothing. So they are constructed from nowhere, you know, but imagine professors, everything, you know, like when people start like writing essays with this, which people are already doing, it's gonna be like crazy times.
I have two quick questions, because I don't know if you have something else to do and we are like over time. But the first question, it's like, if you know any other initiatives to start digitalize all these medical records, because I don't know, but in Latin America, it's not common to have these digitalized medical records. And the thing is that most of the metadata, it's what gives context to this evolution of these diseases. I mean, which is the age of the person, if they have like a clinical history of the family about the appearance of this disease. And it's very important, because most of the analyses that you perform are from public databases, which is already recorded. But from my experience, to apply these to the population that, well, we want to do this at a global scale and all the population, but we need like digital records to do that. And I want to ask you if you know another initiatives to do that, not only in Brazil, but also in Latin America. That's the first question. And the second question, it's more related about, so far, the tools, bioinformatic tools for people that are not expertise in coding are for treating transcriptomic data. But we have so far realized that the regulatory processes in these diseases are not only on transcriptomic level, but for other molecules. That's why we take like a systems biology approach. So my question is, if you have like tried to develop in a near future is bioinformatic tools that account not only to analyze transcriptomic, but to find correlations between transcriptomics, metabolomics, proteomics, phosphoproteomics, and basically of the omics that
yeah, great. Yes, awesome question. Like during the COVID-19, there were like many initiatives trying to integrate medical records, right? So even we have a paper that I didn't show, but we collect data from 10 million records of people doing clinical exam. When you are sick, people ask for a blood exam. You know, these exam usually a physician will look and say, okay, yeah, you have COVID, and throw away this information. So FAPESP collect these 10 million records from different hospitals, integrate this into a database, and make it available. So we analyze and publish a paper on this. When I published the Tukuchi blast, someone in UK said, like, we have this problem, we need to use this tool to integrate that. So not only Latin America, like even everywhere. But the biggest problem, I think, is not only the technological part, it's the ethical part, because that's a very sensitive data, so people are reluctant in giving you this information. We had a meeting with the data suits director like the other day, and they say like, I cannot send you anything. And actually Tukuchi Blast was developed to be like installed in the computer, so they don't have to send anything, integrate there, and then anonymize and then send to researchers. So that I think could be like one solution. The other one, which is digitalizing papers, I know people that are doing, we are also doing this, but I don't know like a big consortium that is doing this. But Einstein, I found out that they have a big data analytics only for this, and I agree with you that that's the major problem. Like most of these medical records goes to nowhere, and they are not used in science. And for the omics integration, as I said, we use already packages such as Mixomics, some Diablo, some tools that do this integration for different layers of information, epigenetics, metabolomics. We have the biofeeds, we had some, I'm not showing, but we have a lipidomics tool that identifies lipids. We also try different technology, but as you said, the most mature technology is transcriptomics. The other ones are but once you transform this into data, into numbers, you know, it's almost the same kind of thing. As long as you have the same distribution, sparseness, if it's a sparse or not, if you want to discretize or not, you know, and after you do this, it's the same kind of approach.
Thank you again for the presentation. It was really amazing. I was going to do the same question as João, if this spatial transcriptomics was real or just science fiction, because it's really amazing. And
this is actually public data, guys. If you want to download that data that I showed you, it's there. The only thing that we did, because I want to invest on this, I asked, get this data, and let's find different ways of analyzing this. And so, but the data is there. And there are some cancer data as well, like for free to download and do it.
The other question that I have is, you showed to us the transcriptomic profiles of some individuals, and you have some kind of clustering if the individual is healthy or has you understood. And I want to know if transcriptomic profile can be a good predictor of disease outcome, if it can be a better predictor than like usual parameters like comorbidities.
So that was my question in my PhD. So my PhD was to identify markers for prostate cancer using transcriptomics. So my goal was to let's use transcriptomics to identify genes that can predict if the person is going to die or not with prostate cancer. And actually, not only my thesis, but thousands of other theses was the same thing, but different cancers. The oncology field invests billions of dollars in screening millions of samples, and so far you have very few examples, successful examples in the clinics that is used to predict outcomes. So that's why none of my papers try to claim we have predictors, we have markers, because we know that that's naive to say that, oh yeah, we identified something that is going to predict very well, because it works really well in your data sets, but if you go to an independent data set, it fails. So that means that it's overfitted. So it means that it predicts really well only your data sets that you use for the paper, you know. So in theory, yes, but I think that for us to get to these markers, we have to get more and more data. And actually, it's not even more and more data. It's instead of big data, is smart data, because sometimes we put more data thinking that we're going to have a better model, but then we introduce a lot of garbage, a lot of noise that affects our models, instead of like getting real beautiful, good data sets to train the models. So there are some companies actually that invest on this. So they go to a hospital such as Albert Einstein and say like, I want your samples of arthritis patients, and I'm going to pay to generate transcriptomics, metabolomics, whatever, because I know that you collect everything the same way, in a nice way, you have clinical information, and then I generate a very good quality data, instead of just going to the repository and downloading 15 different data sets that you don't know the quality, you know? So I think in principle, yes, you can find the markers, but in reality, it's not that simple.
Okay, what I want to ask you may be dumb, and I know it's a problem in science at all, but I believe there are other laboratories in like Brazil and the whole world that do similar things as you.
Oh yeah.
And how do you keep track what tools that have been already created and like what are similar to what you're creating and are going to help you with what you are creating?
Well, the same way you do science, right? So for example, oh, I'm searching for these specific proteins in glioblastoma, and then you find out that someone just published a paper with the same protein, the same experiment that you want to do, and then you say, well, okay, so what we can do now? We can change a little bit of the question and then try to see if someone wants to read that. The same risk that we have when we develop a tool. We usually decide when we develop a tool precisely because there is no tool that do what we want. But in the meantime, someone may publish, and then we say, okay, give up of this tool. It's a normal process. But the semi-tool, the first tool that I showed, was actually entirely based on a very cited tool, which is called WGCNA, which everyone works using this WGCNA tool. But the problem is, for using these methods, you have to write a script. You have to put the parameters by hands, you know. Our question was like, oh, how can we automatize this process? Identify automatically what is the best cutoff, blah, 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 and do all the analysis. And that was the idea of the semi-tool, was not to come up from nowhere. It was like improve a tool that already exists, and that was our goal. But we are always facing this risk of being scooped. And actually, we're trying to make a portal of Brazilian labs that have computational tools to put everything into one website so people can see, oh, I can use this tool, I can integrate, precisely to try to avoid redundancy, because it's hard to read millions of papers, right?
My question is somewhat related. How well accepted are the tools you produce by the scientific community? So how much are the stuff that you're building being used in research?
Well, one way of measuring this is the usage, right? Like we can, in the website, we know how many people went there and do the analysis. Semi-tool, we saw that there were over 100 citations, so meaning that the analysis went to papers that was citing the tool. So that gave us, of course, happiness that people are using and citing. But some, for example, PEBA is a tool that I'm pretty sure nobody's going to cite, because that tool shows the weakness of your data. Because why you're going to cite, say, oh, we use PEBA to decide on this cutoff? No one's to write this. But they can use this, identify the cutoff, and then write in the paper, oh, we use a p-value of 0.07. Do not explain why, because it's arbitrary anyways, you know. But it's fine for me, because I would be glad if people use this. So we were very happy because last year there was a paper from a different group that I did not know, which the title was Semi-tool is better than WGCNA, which is the tool that we use to improve. So the person did a benchmark and saw that our tool was better, and that was like a really nice, you know. But that was not the major goal. The major goal was to develop this tool for the students to do the thesis, because we are not. I'm a biologist. I only make this tool because I have empathy for the people that do not know how to code, So they have to be able to run the analysis without knowing how to program. So I asked my students, so for example, Cesar that did the metavolcano, the thesis was not on metavolcano, it was analyzing arthritis data. So to analyze arthritis data, he has to do the meta-analysis. And when he was doing the meta-analysis, I asked, so why don't you create a package that do the meta-analysis easily? And they say, okay. And then we create the metavolcano, you know, but the products of research was the arthritis story. The metavolcano was a byproduct that if people use, it's going to be nice. So Flavia, people want to use the room?
That's the last one.
No worries.
So I'm a very big fan of tools for public health. And I would like to know, what is the biggest challenge trying to integrate all the records? Because I know that you are already dealing just with the basic data, like names and date of birth. But if you would like to create a tool that could integrate all the data about all the units, what will be the biggest challenge?
I can tell you right now, because it's something that we are already facing. When I download the COVID-19 spreadsheets from the government, they gave me like the information of, one question was, where did you go before you arrived to Brazil that you have COVID-19? So sometimes you found Wuhan, sometimes you found like Italy, blah, blah, blah. And one of the records was Puta que pariu. You know, like, I kid you not, someone wrote Puta que pariu in that place because the problem, the person was talking probably mentioned like a city. The person was like probably stressed, like, oh yeah, okay, this city, okay, and then they typed there. And then we, as a scientist, we saw like this city, Puta que pariu, like what the hell? And one of the biggest challenge for medical records is the free will type. How do you say this thing, like campos livres? Like the person is when the person is free to type whatever they want. It's not like defined by a choice. So if you have to click, oh, you are obese or not, you have diabetes or not, then it's fine. It goes very beautiful to the data. But most of the things person has to type. So for example, when I was analyzing a data set, you know how many different ways you can type vitamin D? 48 different ways. How can you possibly type 48 different ways vitamin D? You can type vitamin D, VTD, VT.D, like 25 OH vitamin D. You know, like it's impossible to create a text mining or a machine learning method that's gonna know what the person wanted. But a human being can say like, oh no, this person wants to say vitamin D. So my challenge is how to convert textual information into defined ontologies. So we can use this to compare and analyze this. Alzheimer's, imagine how difficult it is, how many different ways you can type Alzheimer in SUS database, you know, like, so it's, I think that's one of the biggest challenges for me.
I'm sorry, can I ask just one more question? But let's suppose that we live in a beautiful world where everything works. Do you believe that it could be possible to create this kind of tool and that the community and also the government could accept this in a, like, well? Do you believe that it's possible?
Possible? Yeah, possible it is. But it's also possible that an alien arrives here, yeah, right? Like, but it's unlikely that it's going to be so quick or so easily done. But for example, for this government, it's impossible. I'm hoping that the next data SUS director and then we can have like a more flexibility to really help, because I know that they are like stressed with several other things, like research is the least priority that they want. And that's what we want to help. Like, look, we don't have to pay more people to do the research. Just let us help, and then do this to other people make questions and answer. So we want to do this hardcore part of like processing, integrating everything, and then different people will have different questions and use this information well.
Thank you. Thank you very much for your talk. It was one of the best.
Thank you. Thank you. Falei demais, né?
Fantástico.